The middle fabrics, B cell and T cell zones, and an inner middle. Overall structure includes delicate connective tissue, trabeculae, and an extensive fluid-filled sinus system. Core microscopic architecture. Capsule. Dense connective tissue covering the organ which sends inward projections called trabeculae. Subscapular sinus. The space just the space just beneath the capsule that receives lymph from afferent vessels. Cortex, outer and inner. Outer cortex, B cell zone, contains spherical clusters of lymphocytes called follicles or nodules. Primary follicles are dormant. Secondary follicles feature active germinal centers where B cells differentiate and mature. Paracortex, inner cortex of T cell zone, located between the outer cortex and medulla, populated mostly by T cells and antigen presenting dendritic cells. Medulla, the innermost light staining region. It is composed of medullary cords containing plasma cells, macrophages, and B cells, and medullary sinuses that drain into the efferent lymphatic vessel and the hilum. Next, spleen. The spleen is an encapsulated, highly vascular secondary lymphoid organ. Its histological structure features a dense outer connective tissue capsule that extends inward as trabeculae. The parenchyma is divided into two distinct functional compartments: the white pulp, immune defense, and the red pulp, blood filtration. Key microscopic components. Component key histological features and functions. Capsule and trabeculae made of dense irregular connective tissue, elastic fibers, and smooth muscle. They contain blood vessels and nerves extending into the organ. White pulp appears densely purple basophilic on hematoxylin stains due to high nuclear density. Central arteriole, a small artery applying the pulp, or periarteriolar lymphoid sheath. T cell zone directly surrounding the arteriole. Lymphoid follicles, nodules. B cell zones often containing germinal centers. Marginal zone. The boundary region between the white and red part contains specialized macrophages and lymphocytes. Functions as an antigen filtering checkpoint. Red pulp appears lighter and more pinkish on hematoxylin eosin due to an abundance of red blood cells. Filters aged RBCs and pathogens. Splenic cords or of Billroth, reticular connective tissue meshwork housing macrophages, plasma cells, and RBCs. Splenic sinusoids, wide leaky vascular channels lined by elongated endothelial cells that allow for blood filtration. Spleen, histology. Thymus histology. Thymus. The thymus is an encapsulated primary lymphoid organ responsible for T cell maturation. Histologically, it is organized into distinct globules separated by connective tissues septae, that is trabeculae. Each lobule is divided into a darker cortex and a lighter medulla. Thymic cortex, outer region. Appearance stains dark purple, basophilic due to high cell density. Composition packed with developing T lymphocytes, like thymocytes, and a supportive network of epithelial reticular cells. Function site of initial T cell proliferation and positive selection. Blood thymus barrier found here prevents immature T cells from being exposed to foreign antigens. Thymic medulla, inner region. appearance stains lighter eosinophilic or pink due to a lower cell density and larger cells. Composition contains mature T cells, dendritic cells, macrophages, and unique thymic epithelial cells, TECs. Hassel's capsules, that is thymic capsules, a distinct hallmark feature of the medulla. These are concentrically arranged, tightly packed epithelial cells that are involved in clearing apoptotic cells and regulating T cell development. Function site of negative selection to prevent autoimmunity. Age-related changes in evolution. Thymus is most active during childhood. After puberty, the functional lymphoid tissue progressively degrades and is replaced by adipose fat tissue, a process known as involution. The histology of thymus. Next, palatine tonsil histology. Palatine tonsil is a specialized mucosal lymphoid organ. Its histology is defined by a stratified squamous non-keratinized epithelium, deep tonsillar crypts for antigen sampling, and densely packed lymphoid nodules with a lighter germinal centers. Key histological features: surface epithelium. The exposed luminal surface is lined by stratified squamous non-keratinized epithelium. Tonsillar crypts. The epithelium invaginates into the underlying tissue, forming 10 to 15 deep branching clefts called crypts. These crypts vastly increase the surface area for immune surveillance. Lymphoid tissue. The bulk of the tonsil consists of subepithelial diffuse lymphatic tissue and spherical lymphoid nodules, that is, follicles. These nodules feature active germinal centers where B lymphocytes proliferate. Capsule, a dense, incomplete fibrous connective tissue. Capsule separates the deep surface of the tonsil from the underlying pharyngeal constrictor muscles. Septa extend inward, dividing the tonsil into incomplete lobules. Palatine tonsils. Peyer's patches, installs. Peyer's patches are large, organized clusters of lymphoid follicles found primarily in the mucosa and submucosa of the ileum, the final section of the small intestine. As key components of the gut-associated lymphoid tissue, GALT, they serve as the immune sensors of the gastrointestinal tract. Key histological features. Location. They are located in the lamina propria and extend into the submucosa, the intestinal wall. Lymphoid follicles. They consist of dense aggregates of lymphocytes, predominantly containing pale staining germinal centers where B cells mature, surrounded by darker staining regions of T cells and macrophages. Follicle-associated epithelium. The mucosal epithelium overlying these patches is modified and lacks the typical goblet cells and villi found elsewhere in the intestine. M cells, microfold cells, interspersed within the FAE are specialized M cells. These cells actively sample the antigens and microbes from the intestinal lumen and transport them to the underlying immune cells. No afferent lymphatics. Unlike standard lymph nodes, Peyer's patches do not have afferent lymphatic vessels bringing lymph into them. They drain directly into the mesenteric lymph nodes. The histology of Peyer's patches. Thank you for listening.